Welcome to the Soil CRC's Building Technical Capacity for Improved Soil Management webinar series, evaluating soil functional resilience to compaction and drought. In this video, Griffith University's Dr. Mehran Rezaei Rashti discusses findings from the ongoing Soil CRC research project. The study aims to provide a practical methodology for assessing the resilience of soil in agricultural ecosystems against the environmental stresses of compaction and drought. This research provides valuable insights into the role of management practices and organic residues in enhancing soil resilience to drought conditions.
I'm just trying to make a short presentation about this project that we started to work with Soil CRC. The idea behind this project was that, a lot of different groups and people measuring soil health. So we have many different parameters to measure soil health, but the problem is that the results are usually not consistent, and it's really difficult to adopt it in farming systems because they become so expensive. So in collaboration with Soil CRC, we end up with an idea of evaluating soil functional resilience to environmental stresses, in this case, the compaction and drought, which are two of the main environmental stresses in Australia. We wanted to have a mechanism-based understanding of how these stresses affect on soil health, and eventually, hopefully by the end of this project, be able to reduce the number of the tests that we have to do and reduce the cost of sampling and analysis of soil to end up with a simple, robust, and applicable soil health package test for different farming systems to be able to adopt it in their own farming system, their own paddocks, with minimum cost and maximum, basically, value. So we end up with a project, we call it Evaluating Soil Functional Resilience to Compaction and Drought Stresses for Developing Higher Performance Soils. So in these projects, we have researchers from Griffith University, New South Wales DPI, and we also work with two farming groups. One, Herbert Kane Productivity Services from Queensland, which I see Richard is representing them here. And also the FACEI group from Western Australia. So the FACEI groups mainly deal with the drought problems, and basically Queensland farmers mainly have issues with compactions. So we try to put them both together and have a proper mechanism-based understanding of how the soil reacts to environmental stresses to be able to end up with some solutions. So some background before I show you some findings of the projects. As you know, the definition of a healthy soil is a soil that have the continuously capacity to provide optimum amounts of nutrients, water, and air for growing plants and provide a disease-free environment for the cropping systems. But due to over-application of mineral fertilizers and conventional management systems that adopted to different farming systems, we end up with a continuous decline of the yield and economic profitability of the farming systems, which mainly associated with reduction in soil fertility and health due to different environmental stresses and basically adopting of different farming systems. But if you want to make a change, first we need to end up with a simple, robust, and affordable method for farmers to test their health status and give them basically farm-based or paddock-based recommendation to be able to improve the health of their soil and improve the fertility of the soil. In order to do that, we first have to have a good understanding of the process that the soil goes through when they're dealing with the environmental stresses. So if you look at the graph here, I'm trying to simplify what happens when the soils goes through different environmental stresses. So they have two definitions here. The first one is soil resistance, and the second one, soil resilience. So for example, if you're at the top of the graph, your normal soil in the paddock, when you're dealing with a different environmental stress, such as drought and compaction, the functionality of soil, physical, chemical, and biological properties start to degrade. So the capacity of the soil to deal with the environmental stresses is called the resistance. So the soil start to resist against the changes that force through the system by different environmental stresses. But what happens if the environmental stresses becomes long-term or so frequent to happen? Then the soil cannot basically resist against the changes. So the soil have to change. Physical properties, chemical properties, and mainly biological properties start to change when the stresses become more frequent or so long-term. So as you see, the basically functionality of the soil start to decrease when we're dealing with these kind of stresses. But what happens when the stress is lifted, when we remove the stresses, when we do plugging, if we have the compaction problem, when we irrigate the soil, when we have the drought problem, or we have a rainfall. So the soil start to restore its functionality, physical, chemical, and biological. The capacity of the soil to recover itself and get as close as possible to its original functionality is called resilient, which is the basically main point of these projects that we try to improve. If we end up to improving soil resilient, we can make sure that when we have environmental stresses in our systems and the environmental stress is lifted after a while, we have a higher chance to recover our soil functionality to its close to its optimal condition. Why is it important? Because as you see in the graph, every time that your soil goes through different environmental stresses, lose parts of its functionality. And when it recovers, it doesn't recover to its optimum condition. So it loses its functionality after each time. If this process happens over a really long time or more frequent over a long time, eventually you end up to a status that the soil will irreversibly damage or permanently damage. So the functionality will go so low that it cannot recover itself. And that's the point. Then we started to get soil degradation. We started to lose the profitability. We start to lose the crop yield. And the cropping system is not profitable for farmers. The problem here is that if the proper profitability goes down, then our capacity to improve soil health by applying different amendment goes down, because there is no profit in the system to be able to use to improve soil health. So we don't want to get to that critical threshold point. Before we get to that point, we need to do something to the soil as long as we have enough profitability to reinvest into the soil system. So these projects try to have an optimum understanding of the mechanism-based processes that happens in soil microbial community composition and diversity, soil chemical properties, when the soils go through drought and compaction stresses across different farming systems, and end up with a model that can predict soil health status, and then simplify that model to a couple of simple tests and a couple of cheap tests that's affordable for every farmers, every farming systems to test their soil health and improve it. So the expected outcomes of these projects is broad application of the better management practices through different projects that going through Soil CRC, different farm-based projects, and also for a broader Australian agriculture industries. Here is an example of how we do our incubation experiments. Before I go further, I need to mention that these experiments are lab-based experiments, because the idea behind these projects just to find a mechanism-based understanding of the process to help us to get our finding to the farming lands and to the broadacre cropping systems. So the experiments that we do here are all lab-based experiments. For example, you see here, we have a jar that we put the soil in, and we use our compactor machine to compact the soil to different compaction systems. We also introduce different droughts to the samples, and we look at their functionality. So these projects try to develop different assessment methods and management protocols to improve soil functional resilience against the stresses. Why is it important? For example, if you look at the graphs on the right-hand side, when you have a resilient soil, you have reduced the amounts of your carbon loss, either through leaching from the subsoil or through basically CO2 emission or greenhouse gas emissions. And also you can keep the integrity of the soil intact. Why is it important? Because in a good year that you have enough rainfall or a good condition in your farming systems, basically most of our soils produce similar amount of yield. But in a bad year that you don't have enough rainfall in the system, the soil is not in its optimum condition. A resilient soil can guarantee a minimum yield and can guarantee that you don't have too much loss in your profitability, and you can safely basically through those bad years and move on to the next couple of years, which probably you get a better condition by changing the environmental processes. So how we do that? We provide different stresses to the soil, and we assess soil functional processes and microbial community composition and diversity, and relate them to the soil fertility and plant productivity. We also try to provide practical methods using innovative biochemical methods for the engineering of target soils against these different environmental stresses, and also providing soil resilient protocols to the other project leaders of Soil CRC to be able to test it in different cropping systems and basically modify it based on the requirements of the different cropping systems. Also, we do international collaboration through basically providing different papers, journal papers, or presentation in different conferences. So what we have done till here, we had different meetings with the research providers and the farmers community and farming cropping systems to design different experiments. We selected different field sites, both in Western Australia and North Queensland, under different gradient of management practices for our experiments. We're in the process of providing a literature review paper, which will be useful both in Australia and worldwide to have a better understanding of the resilience of the soil against different environmental stresses, which is not a really simple concept when you bring it to the broadacre systems, and also providing different scientific papers, technical reports, fact sheets. But just remember, this is a lab-based experiment, and we're not applying anything to the broadacre field systems at this stage. So what happened till now? We finished a literature review of the projects. The PhD students that has been involved in these projects had a thorough literature review regarding the effect of soil compaction and drought on basically soil health and improving the resilience of soil when they're dealing with these problems. We finished the first experiments of the project, which was basically 72 days incubation experiment. So for this experiment, we picked two soil types, a clay soil and a sandy soil, two levels of soil moisture content, 15% and 35% water-filled porous space. Just need to remind you that water-filled porous space is different from water holding capacity. When we're talking about water-filled porous space, we're mainly talking about the amount of the soil pores that filled with water. So it's different from how much water can be held in the soil. It's mainly a combination of the compaction and availability of water. So when you put them together, it becomes a different factor. We call it water-filled porous space, which means how much of the porosity is filled with water, which decide if your system is aerobic or anaerobic, which really helps in interpreting your result, because mainly when you're talking about the biological systems and soil microbial activity, it's really important to know your systems is aerobic or anaerobic and what kind of organisms are active in each condition. We also used 6 different levels of compaction, from a really low compaction or 0.9 gram per cubic centimeter up to 1.5 gram per cubic centimeters, which is a severe compaction issue in the farming system. So we tried to identify the main indicator of soil resilience. How we did that? We measured a combination of different chemical and biological properties. Also measured the diversity of soil microbial communities to be able to identify the most important factors and have an understanding of the mechanisms that are going around, to be able to later on simplify the process and also introduce the application of organic matter and see the effect of the system or change on the system when you introduce an organic matter. So the first incubation study was based on understanding of the mechanisms without improving soil condition by application of organic matters. So the findings for these experiments indicated that soil microbial metabolic activity, as measured by the respiration or enzyme activity, responded differently to different levels of the drought and compaction, and also were really different on different soil textures. So to have a good understanding of what I mean by microbial metabolic activity, I can explain that, for example, assume that your soil profile is living like a human. So for example, the soil profile is living like me. So generally, I respire more CO2 than you guys because I'm fatter, I'm heavier, I have more body mass. So when I have more body mass, it means that my metabolism is faster, my metabolism is higher, so it means that I produce more CO2. The other reason that I can produce more CO2 is when I'm running, when I'm active, when I'm doing something. So my body is under stress, so my metabolism goes higher. So although I have the same body mass, but I'm respiring more CO2 because I'm under stress. So what happens here, we try to differentiate between the amount of enzyme activities and CO2 respiration into the soil system when the soil is healthy, and the reason for more enzyme activities and CO2 respiration is because the soil is producing more microorganisms and we have more mass and we produce more CO2. Or when we produce more CO2, the reason is because the soil is under stress, our microbial community are fighting for their life and try to generate more CO2 because they're more active. So what we have done, we had our 2 different soil types, a clay soil and a sandy soil. We put the soils under different stresses of compaction and drought. The first step of the experiment was to apply the stress. As you can see here, we applied the stress at the beginning of the experiment, and we monitored the changes in soil chemical and biological properties to see how they degrade when they get affected by environmental stresses. For example, here the graphs that you're watching is related to the extraction of hot water extractable organic carbon. This is slightly different from extraction of organic carbon. In this case, we keep the soils in hot water, 70 degrees centigrade for around 16 hours, and then we extract the samples. Our previous experiment shows that this is higher relationship between the extracted hot water extraction of organic carbon and your available food for microbial activities. So when you're measuring hot water extractable organic carbon, you have a better understanding of how much food is available for your microbial activities to grow. As you see, the pattern is slightly different when you're comparing a sandy soil to a clay soil, and also when you're comparing a high moisture condition to a low moisture condition. But overall, in all of the graphs, when you put the soil under stress, your food availability or hot water extractable organic carbon availability decreased by time. When we left the samples for 35 days, we decided to release the stress. So if it was under drought stress, we add water to the system. If it was under compaction, we remove the compaction and to see how microbial community react. So for example, in this case, we release the compaction and add water to the systems, and you see the hot water extractable organic carbon start to increase. So the food availability start to increase in the systems, means that the composition of organic matter is accelerating, but you see there is a gap between different compaction levels, and there is a gap between recovery of the soil systems after the releasing from the stress. So the process is different from sandy to clay soil, and the process is different from low moisture condition to high moisture condition. Similar things happens when we look at the respiration of different soils in different conditions. As you can see, when the soils start to go under stress at the beginning of the experiment, the difference between treatments minimized because all of them have been under stress, all of them been reducing their functionality. So the CO2 respiration decreased as a result of reducing the number of microorganisms. But when you release the compaction stress, they completely react differently. Some of them recover really fast. In this case, the one with the low compaction. So, and basically higher moisture content, especially you can see here, if you have high moisture content in a clay soil, the recovery rate will be completely different comparing to the time that you have low moisture content. And the same happens if you're looking at a sandy soils, the recovery rate is completely different. So this will give us some idea of how we can basically relate different analysis systems, which are really cheap. For example, here measuring CO2 emission is really cheap. So you see that we can capture the difference between recovery of the soils using a simple test like CO2 emission. But the trick is here that we need to relate this simple test to different properties of the soil, biological property, chemical property, and physical properties to make sure that the result that we're getting is a true representative of the soil. And that's why we measure a lot more different parameters to find out if our result are robust or explainable or not. For example, here we're measuring one of the main enzymes that's responsible for external digestion of the food. You know that the microorganisms cannot ingest the food inside their body. They need to digest the food outside and then basically consume the digested food. One of these enzymes, which is external exudation of soil microbial communities, is beta-glucose, which is effective in soil carbon cycles and effective in decomposition and release of carbon in the soil systems, you see we have the same pattern. When the soil goes under stress, the exudation of beta-glucosides decrease. And when we release the compaction, we have more enzyme activities in the systems. And these enzyme activities are different when we're dealing with different compaction levels and different moisture levels, and also in different soil types and soil textures. Next one, the changes in soil microbial community composition. So if you look at the left-hand side in A, that's our soil that has been under no stress, so it's optimum condition. And in the left-hand side, you see a soil that has been under compaction and drought stress for 35 days. And you see that the community composition of soil microbes are changing, and it means that the functions of soils are changing when it goes through different environmental stresses. One example, which is a really simple example, if you put your clay soils, which is really common in North Queensland, when you put your clay soil under compaction stress, you reduce the amount of CO2 that's produced, comparing the soil that has been under no stress. So it means that the microbial community comes down, less CO2 production in the systems. But because the systems change from aerobic condition to anaerobic condition, the anaerobic microorganisms start to increase, and your nitrous oxide emission increased by 5-fold. And this is one of the other issues that we have in North Queensland, which we lose a lot of nitrogen through greenhouse gas emission, similar to runoff, similar to deep drainage. But the changes in the compaction not only affect the carbon cycles, but also affect the nitrogen cycles and nitrogen use efficiency. And that's related to the change of microbial community composition in aerobic and anaerobic species. After we finished that experiment, we went to the second phase of the project, which in this case, now we understand the limitations that we have in our cropping systems. We know how the systems works when it goes under stress, and we know how the system reacts when it gets out of the stress. Now, in the second part of the project, we try to introduce crop residues to the systems and find out how the crop residues will affect and improve soil resilience against drought and compaction. So this is the part of the projects which is already in progress. So for example, we designed 2 different experiments. One experiment was mainly focused on drought, and the other experiment was mainly focused on compaction. So this experiment you see here is the drought part of the experiment. We have 12 treatments, and we collected 2 different soils adjunct to each other. So they have the soil structure, the same soil texture, and basically the same nutrient profile, but they have been under different management practices. So the topsoil has been under conventional management practice for a couple of years, and the bottom soil from treatment 7 has been under best management practice for a couple of years. So we have 2 different soil types, 2 different soil adjacent to each other, same texture, different management practices. The same as previous experiment, we put them under stress and to see how their functional properties change when they go under stress. Is the historical management practice has any effect on changing the mechanism when the soil go under stress? And then after a while, we remove the stress. For example, in this case, we remove the drought, and also we apply organic matter content. So here you can see when we lift the drought, we had 3 moisture condition, 15% water holding capacity, 30% water holding capacity, and 55% water holding capacity at the beginning of the experiment. But when we lift the drought, we remove them, we bring them up, all of them to 55% water holding capacity, which is the optimum condition for plant growth. And also for half of the treatment, we apply the organic matter into the system. So this organic matter has been chosen based on the available organic matter in different farming systems. We apply to the systems, and the same process, we look at the mechanisms of the recovery of different, basically, treatments without application of organic matter, and the one that has been under stress, no recovered, comparing the one that hasn't been under stress into the system. And that's it. So I'm happy to take any questions. So when you're looking at the microbial activities, you need to separate their activity based on the optimum condition and their stress. So sometimes you have higher microbial activities. If you're looking at their mass, it's a good representative of their activity. So when they have higher mass, they have higher activities. But measuring the mass is usually more problematic and more expensive. So the easiest way is to see the respiration. But the problem with the respiration is that you need to differentiate to see if this respiration is coming from the optimum condition or it's coming from a stress condition. So any products available commercially can be easily calibrated against the standard methods. And when you calibrate those products, either it's for measuring the microbial activity or measuring the respiration, when you calibrate it properly based on a soil type and basically the methods that we use in the labs, and you calculate the amount of error, then you can accurately use them in your farming systems. Really cheap, really effective, but already calibrated to the optimum condition.
Thank you for watching. If you found this information useful, there are many more related projects to discover at our website. Please like this video and subscribe to our YouTube channel for more Soil CRC webinars, project updates, and conference presentations. This webinar series was produced by the Soil CRC, jointly funded through the Australian Government's National Landcare program, to build the technical capacity of natural resource management agencies, Landcare and grower groups to deliver soil health information to farmers for improved soil management. For more information about our projects, visit soilcrc.com.au.